for millions of people. And mental health is taking hits in different people in different ways. And just want to make the point that breaks, vacations matter. If you're a student and you need time, be sure to discuss with your PI, make appropriate arrangements. And remember that psychological services are available through the university. And it's easy to find this with Google, psychological services, Wayne State University, and you'll end up finding this quite quickly. And an important thing is don't wait to ask for help. So if you're having things where you're not sure, where some help could be useful, and I think this actually is worthwhile, that don't wait too long to ask for help, okay? We're going to talk a wee bit at the very beginning and then not come back to this very much through the rest of the talk, that this virus, this pandemic, everything associated with it is extraordinary behind anything that's happened in my lifetime. My dad's 96. He wasn't around for the pandemic 102 years ago or so, which is the last time something of this global nature has really gone on. I grew up in the period when vaccines for early life virus infections were just happening. Polio, measles, I got measles, mumps, rubella, and chickenpox myself, and took some of the early oral vaccine that was developed by Dr. Sabin. We walked down to my elementary school a Sunday afternoon to get little cups of sugar water with that. So those were extraordinary periods when everyone was susceptible to the virus, but it didn't hit everybody all at once in the way this one is. This virus is also a very dramatically pathogenic virus, and it's doing things, multiple systems in bodies, and it's doing it quickly, and it actually does a whole lot of destruction of tissues and then triggers responses that cause further pathology. And for reasons that we'll talk about, this is likely to go on for some time. And so this is a real society-changing event. And so that creates, I don't want to say this is fun opportunities, but there are even responsibilities that some of us have that go in the direction of doing things related to the disease, coronavirus disease, starting in 2019, COVID-19, and SARS, severe acute respiratory syndrome, coronavirus number 2. Number 1 was the 2002-2003 epidemic, pandemic. And so at the university level, there are people who are in the business of telling stories in various ways, photojournalists, written journalists, artists, musicians, a variety of other ways for telling the story that can engage us. There are legal issues. Working in collaboration with Dr. Jeff Ram and thinking about looking at sewage to see whether there are transmission patterns or ability to detect virus on an early side and then detect waning of virus activity in sewage, and actually required some legal analyses now recently published on that front. And there are other things that can connect to legislation and other areas of interest within the university. There are socioeconomic disparities that end up connecting to healthcare disparities that need to be documented, and at least as important as documenting them is trying to find solutions for them. We've got things on the clinical front, and there are a lot of people at the medical school engaged in this already. Improvements in diagnosis, speed, specificity, sensitivity, and treatment. And there's treatment that affects the virus itself, controlling the virus, as well as treating specific pathologies. We have public health epidemiology that needs to be done. Who in the population has gotten the virus, what's happening over time at the population level, and this important thing of case tracking, where we're actually a bit behind the 8 ball and not having gotten this started soon enough. But in order to control epidemics and pandemics, case tracking, monitoring who made contacts and how is the virus moving so that steps can be taken to control that is extraordinarily important. Prevention, use of vaccines, vaccine trials. People at the university will be involved in those. Behavioral interventions, social distancing, how do you motivate it? What forms of education, communication in different populations actually work at leading to measurable prevention. Antivirals, I'm moving more toward the chemistry direction. Antivirals. In order to develop antivirals that you can test on the virus, this is a virus that is very contagious, it's very pathogenic, and most of the population is susceptible to it. That puts it right now at a biosafety level 3, and I've heard that there is discussion of whether it ought to be at a higher containment level. This puts constraints on things we can do at Wayne State because we don't have that sort of high level containment facility. Nonetheless, things can be done. There are some sorts of things where biological specimens can be processed so they can be worked with them safely. And then there are surrogate systems like pseudotype viruses. And Dr. Nguyen from the chemistry department's working on some things that connect in that direction. And then working on other antivirals. Dr. Ladislao Kovari in our department's working on protease inhibitors. And then there would be not the virus specific part of it, but actually the pathogenic part of it, drugs and therapeutic approaches for treating the diseases produced by the virus. So many, many opportunities, more than I outlined here. And I did this as much as anything to emphasize the scope of the impact that this virus has had already and is going to continue to have for years to come. Some of the things that we're going to talk about, again, I said the name of the disease is one thing. We have a different name for the virus. Sometimes they get sort of conflated in sentences and it can be a little bit confusing. We're going to spend a few minutes talking about basic virology of coronaviruses, their biological properties, their physical properties, and how that contributes to the pandemic. We're going to talk about a concept I got from a neighbor who is a chemical engineer and has management responsibility for a group that's stayed active during the pandemic because they provide feedstocks for industrial purposes. And he told me their concepts of layers of protection. I think this is really useful. We'll talk about that. And then a little bit later in it, we'll talk about some diagnostic tools and the various ways they might be used so you understand some of how these things might be being discussed and what things might be relevant to you, both in clinical and in pandemic management, paths for treatment and prevention. And then I imagine almost everyone on this video conference has heard of the magic thing R0 or R0, some call it. And I want to actually spend several minutes talking about this, what it is, its characteristics, and why we need to work to keep it a small number. A very brief historical review going back to March 22nd, where this is on the left side here, we have the number of cases in the US. This isn't international, almost 300,000, 127,000 deaths over here between mid-January and March. By April 14th, here are the graphs from then. They actually look quite similar, except the scales changed so that this number is now down here. And in that period between March 22nd and April 14th, that we had a significant increase in number of cases as well as numbers of deaths. Getting to yesterday, that April 14th number is down here, the death number is down here, and in the period, just that handful of weeks, that deaths, new cases keep going up. And it's worth understanding also that these are the counted numbers through public health departments, state departments of health and such, and there's a lot of good reason to understand, not guess, but know that ascertainment bias is happening. That's undercounting. And one analysis I saw recently, it was in the vicinity of 25 or 30% in the US. That is, this says that there are this many cases and there are probably 1/3 more actual cases. Likewise, actually with deaths is what they were specifically counting. Let's look internationally. We're up to over 6 million known cases. I mean, these are active cases. We got that many deaths, 400,000 almost, and almost 3 million people recovered. That's 6 plus 3. We're working toward 10 million cases internationally. And again, these are the counted cases, and there are parts of the world, South America, Africa, Russia, other places where undercounting is actually pretty clearly documented right now. So the actual dimensions of the epidemic are much more than we get out of the numbers that have been carefully counted. The time course. If you get the file that has the links, this is a good one to look at, watching the time course happen in a video sort of way. I've just pulled out particular dates. January 6th, there's a case reported in Seoul and in Bangkok. We get 4 weeks later, and we're starting to get the eruption in China, a few cases to North America and Europe. That's this date now. 4 weeks after that, we've gone wild in Spain and France and Italy and in Iran, and it's starting to erupt in New York City. 4 weeks after that, we've had the big explosion in New York, big cities predominantly, as well as elsewhere. And that's that number. And then we go to May 31st. Between April 6th and May 31st, the graphs look quite a bit different. They had to change the scale because the numbers got so big. This would just be one big blotto of blue for the most part. So this thing is changing rapidly on an international scale. So the disease, much of it happens in the upper respiratory tract. Lung infection leading to transmission via coughing, church choirs, singing. That's my parents are into that stuff, and they're not able to have those events now. Just speech, breathing, intestinal infections. This virus infects intestinal epithelial cells, leads to presence of the virus in sewage at a level of, this is phenomenal, over 10 to the 7th, that's 10 million per liter. That's 10 per microliter, 10 genomes per microliter in Paris sewage. This is a phenomenal output of virus going that far. Sites of infection, many cells that are protected by mucous membranes throughout the body. It seems to like epithelial cells, I think. And the range of disease can go from, we've all heard of asymptomatic or very mild infections. A lot of times people think they're asymptomatic, but on careful and questioning, turns out, oh yeah, I did remember something like that a couple of days ago. And then the fatal infections, asphyxiation, clot-related disorders that can lead to loss of limbs, strokes, heart attacks, pulmonary embolisms, multi-organs can be involved, and cytokine storms where the immune systems run a bit amok and can cause severe health problems. We're still in the zone of, this is yesterday numbers, still 20,000 new cases per day in the US. So we're going to talk about this as a virus, and I was a chemistry major way back when, and it took me a long time, even after I became a graduate student studying viruses, to begin to think about what's the difference between virus and bacteria. And I want to make that point clear today. So viruses as a group, and there are 1.5 zillion viruses, I think is the actual count, that they are metabolically inert on their own. They can't do very much. They are infectious, we know that. Here's a key word, a key phrase. They are obligate, it must happen. Intracellular, things happen inside of cells, and they're parasites. So viruses are dependent on cellular systems. They have to do their thing inside living cells. They co-evolved with cells. Virus genomes are either DNA, RNA. The new virus is an RNA genome, and the virus genome has all the information necessary to direct its replication in a cell using cellular systems, ribosomes, transport systems, metabolic systems, endosecretory apparatus in the appropriate host cell that the virus evolved to do its thing in. The virus particles, the thing that moves through space to infect one person from another person, or one cell to become infected from another cell, that these particles are called virions. They're not viruses. The virus is this overall entity. Virion is the package that's formed in the infected cell from new parts new components within the host cell, and that virion then is the vehicle for transmitting the virus from cell to cell, be it cell in one host to cell in another host to the next host cell or organism. And then it attaches to a cell, it does a little disassembly to deliver its genome into the cell, and that initiation, the disassembly inside a new targeted cell is when the beginning of the next infectious cycle begins. So this is the second and last of these what's a virus slides. So they are biomechanical devices. I can think of them very much here in Detroit as many, many analogies to automobiles and the manufacture of automobiles. They have lots of little parts. Parts need to make sub-assemblies. Things need to be delivered to particular places in cells in a particular sequence, so just-in-time delivery of parts. The order in which you put the parts together matters. If you put the body on the car before you put the seats in it, it's hard to get the seats in. And viruses are associated with all forms of life. There are viruses of bacteria, there are viruses of fungi, there are viruses of viruses. And humans are host to several hundred different viruses. So it's genetic information in a deliverable package. People can argue around campfires, are they alive or are they not alive? But we talk about growing viruses. The minimal virus is the genome. It's got to have instructions on how to make more of it, and a coat of some sort. We'll come, not going into this in depth. And again, they need cell systems, and one of the ones that really helps me understand the difference between virus and bacteria is that they are translation parasites. They must make use of cellular ribosomes. So a major distinction from bacteria, where if you provide the appropriate nutritions and environment, many bacteria are able to divide and grow just by feeding them properly. And viruses, it's not a question of put them in the right media, it's a question of allowing them access to the correct cell type that they're able to enter, have the appropriate binding receptor in molecular interactions, and then for everything else to happen inside the infected cell in a way that took millions of years of evolution to enable. So the virus, SARS-CoV-2, is a coronavirus. We've heard that. There are funny songs related to it and stuff now. Coronavirus, virions, when we look at them through the electron microscope, have a halo or a corona. Looks like a crown, the corona. And it's made up on the virion surface, virion's spherical. On its surface, there are some spikes, and I'll show you pictures in a minute. Those spikes interact with cell surface molecules, the virus receptor, at the step of virus entry. So the spike protein of SARS-CoV-2 is embedded in a lipid bilayer that's derived from host cell membranes. It's what we call an enveloped virus. It has a genome of about 30,000 bases, not base pairs. It's single-stranded RNA. The RNA is able to function as a messenger RNA or an mRNA that can drive the expression of 27 or 29 proteins. So the genome itself is able to interact with ribosomes and direct the translation of proteins. I'm going to show you a picture of this in a moment. So let's move on to that. Now, for most audiences, other than chemistry department, maybe some people in physics, I can say virions are small. It ends up, that's a small joke when I talk to chemists, because the molecular weight of SARS coronavirus virions, my sort of guesstimate, is in the zone of 40 million. The genome's about a 10 million molecular weight, and then we'd have the various proteins and lipids and stuff associated with it. But let's stay with this. Virions are small. That's a red blood cell. A lot of us have looked through a microscope at red blood cells, added some water, watched them explode, or put some salt water on and watch them shrink. Relative to that, which you can see with the microscope in high school laboratories, that there is an E. coli bacterium. It's a living cell. Feed it the right stuff, it'll grow and divide. And the little specks down here are actually viruses, virus particles, virions. Zooming in, we see those. Smallpox virus, it's also known as variola or variola virus, is one of the largest physical virions of viruses of humans. That out of the way. Bacteriophage T4 has a genome that's bigger than the genome of the coronavirus, and it's stored in this head, which is sort of like the size of the center of the coronavirus virion. There's some other viruses, poliovirus, much smaller genome, much small virus particle. You can see we get down from here and we see bacterial ribosomes are smaller than any of these entities, but we're in that sort of ballpark size-wise. We can make a lot of virus in culture in laboratory of at least some viruses. So 1 teaspoon of some laboratory grown virus stocks, and I think that this virus is in this zone and it's on the hot side of it. 1 teaspoon, which is about 5 milliliters, can hold 5 billion or more infectious virions. Okay, so many more than there are people in the United States. And I've seen things to suggest that this virus actually can probably produce over 10 to the 9th in the direction of 10 to the 10th infectious units per milliliter in culture, which is a large amount of virus. When we think about viruses, not going into this in great detail, but out of all the viruses that we classify them, this helps us understand things about new viruses that are related to other viruses. And we can classify them based on their genome. Coronaviruses are RNA genomes. We can classify them on the nature of their capsid. They have one we call helical. You'll see why in a couple of minutes. And we can classify them further on if they have an envelope, a lipid bilayer, or they don't. So we have an RNA virus. It's got a helical genome. It's in an envelope. And this column here gives us some of the details about coronaviruses. It's a plus strand virus. That means its genome is able to operate as a mRNA. And it has the largest genome of any of the RNA viruses in this table, about 30,000 nucleotides. So what do they actually look like? These are pictures not of the new virus, but of the 2002-2003 virus. And these were the first pictures that came early during that epidemic that let everyone know that there was a coronavirus. So a couple of friends from my CDC days took these photographs, and just to humanize it a little bit, the person that took this photograph, one of my daughters used to babysit her kids. They lived just a couple of houses away. And I've got a picture of a herpes virus in my wallet that whenever I need elbow room on an airplane, I can say to the person next to me, hey, you want to see my herpes? I can't hear you laugh. Turn your mics on. Okay. So these roundish things are individual virions. And we can see this little halo around here. This one, quite nice. These are thin section where the 3-dimensional virion was sliced into very thin slices, and we're just looking at little discs of it, so we can just see a little bit of hint of the halo around the perimeter. This image is done where we've got the 3-dimensional virion and it goes on a grid, and we take a picture of that and we get more 3-dimensional information in the image. So we can see the capsid that contains, or the main part of the virion that contains, this is enveloped and it's got the genome inside it. And then we can see these bulbs on the outside. We'll see some other representations of these. These are the famous spike protein that's on the surface of the virion and does the interaction with target cells at the entry stage. So we can see very nice, the corona that way also. So going slightly more anatomically correct, from the inside out, we have the genome, the green line, and it's coiled around this way, and it's associated with nucleocapsid proteins, and it packs very tightly and compactly at the core of the virion. We have a lipid bilayer out here, and it's got a number of virally encoded membrane protein, brown ones, and small membrane protein, these orange ones. And then there are trimers of the spike protein embedded in the membrane also. So virion molecular weight in the vicinity of 40 million. And on the scale of viruses, these have, I think, more environmental stability than herpes viruses and less environmental stability than polio viruses. So polio viruses don't have envelopes, and they evolved to be ingested orally, to replicate in our digestive tract, and to be excreted and ultimately to get into waterways and survive days of being in water, sewage, other things, to then be consumed by someone downstream and establish the infection. So poliovirus virions are extraordinarily stable environmentally. These aren't quite that stable, but they're pretty stable on the scale of envelope viruses. And this actually tells us some things. This is a lipid bilayer. What do you use to dissolve lipids? Talking to a bunch of chemists, I know they can't hear you, but I know you're all thinking detergents, ether, things of that sort. And that's absolutely the right answer. Nicely done. So a little bit more anatomically correct. A different representation, but here we can see the lipid bilayer. We can see both sides. We can see the long chain hydrocarbons extending down in there. So we got a good lipid bilayer here. Inside we have the coils. The pink would be the single-stranded RNA genome wrapped in coils associated with the nucleocapsid or the nucleoprotein. And then those coils are coiled together in a very space-efficient manner inside the virion lipid bilayer. And then we have spike protein trimers studding the surface and the other glycoproteins also on the surface. And the glycoproteins are interesting. And I know Dr. Nguyen and some other people in the chemistry department work on glycopeptides of various sorts. And many of the representations of the spike protein, just for convenience, don't include the sugar groups that are part of it. But we're getting some nice images now of the complex glycosylation patterns. These are just different views of the spike protein primer. And these fuzzy zones on it are different forms of sugar groups that have been attached to it. And this actually is the biologically relevant form of the spike protein with its full glycosylation. This is what the immune system sees. And the question about all of these is, what are their effects on vaccine performance, and how might this glycosylation be modulated in the course of producing vaccines to potentially enhance performance of the vaccine? So now we get to the genome. We're just about done with the molecular virology part of things. This is the genome. It's just a line, and the boxes along it show open reading frames that can be translated into the 20-some different proteins. 3' end has a poly-A tail. 5' end has a cap that's able to interact with ribosomes. It's a eukaryotic mRNA, able to interact with ribosomes and start translating. It's an unusual RNA. It's 30,000 nucleotides long. Its first translation product, there's actually 2 of them that can happen. It starts to translate, and it can terminate here, and it gets 1 polyprotein, or it can do a little ribosomal frame shift right here. So there's some interesting molecular mechanisms involved in that. And instead of finding the stop codon that's there, it can actually shift reading frames and continue to a stop codon down here. So it ends up translating this much longer polyprotein. This is about a 7,000 amino acid protein. Embedded in that protein sequence are 4 enzymes. There's a helicase, there's an RNA-dependent RNA polymerase, but they aren't active when they're in the polyprotein. They need to be cleaved out very specifically. And so there are 2 proteases that are part of the polyprotein that are they're active even while they're in the polyprotein. This one, the 3CL, clip, 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 but the blue triangles liberates these proteins, and the PL proteinase liberates these other proteins. So virus genome encodes at least 4 enzymes. I say enzymes because that makes them potential antiviral targets. 2 proteases. Dr. Kovari in our department is a virus protease specialist, and he's working on developing drugs that target that with some people at Emory that are world-class antiviral developers. Working on and there's a helicase, and there's the RNA-dependent RNA polymerase. This is the enzyme necessary to replicate the genome and to make both the plus and minus strands that are needed. I'm not going into details down here, but during the molecular gymnastics that the RNA-dependent RNA polymerase and the helicase, and I imagine some other proteins are able to, that are present in the virus that it ends up being able to make subgenomic mRNAs that are capped, have polyadenylation, and each of them translates one and sometimes two proteins, the one that's most N-terminal to each of those shorter mRNAs. So a fairly complex replication cycle in order to get all of those mRNAs generated. You've probably heard things about the origins of the virus. Did it come from an animal called pangolins? Does it come from bats? I don't have a good answer for that. It's clear that there are many different species of bats, and closely related bat species can share some viruses that end up evolving the flexibility to infect closely related hosts. There are some animals where bats can infect camels, and sometimes out of that emerges a virus that's able to infect humans, Middle East respiratory syndrome coronavirus. The new virus, it's not entirely clear. I'll tell you what the most recent information I know of, which is about 2 weeks old now, so it may have changed some. It's been moving rapidly. So across most of those 30,000 nucleotides, the most closely related virus is this bat coronavirus, TG13. There's about 96% identity across the 30,000 bases between that virus and the new virus. 4% of 30,000, 1,200 or so nucleotides differ between these two viruses, and across the genome, that the variation actually, it's not even across the genome, but there is variation across the genome, which says that it didn't go from being this virus of bats to become a virus that seems to be pretty happy to be in humans and able to persist in populations now in a single evolutionary jump. So there's some mystery meat still in exactly where the virus came from. Okay, moving in the direction of safety-related things and concerns. So we don't always know when we sneeze, but if you take the picture with the right sort of strobe and the right kind of backlighting, that a lot of stuff can be put out in the course of sneezing, and it can move pretty far. When we think about airborne transmission of viruses, there's actually a gradient here between what's called droplet and airborne. And Ebola virus, which is a hemorrhagic fever virus, and it leads to blood leakage. And when it does that in the lungs, when people cough, what they're putting out are fairly large blobs of bloody material that fall out of the air and don't go very far before they hit the ground. Just gravity is able to get them. So large droplets fall from the air, and that's a function of the form of pathology of Ebola virus in the human lung. This is why when sometimes individuals have started off the day on some continent at a distance from the US and gotten on a plane and been on a plane for 12, 14 hours and gotten symptomatic with Ebola while they're on the plane, that there are no cases of transmission of Ebola to passengers on an airplane where someone who was incubating the virus was on board. And one of the reasons for that is likely that had they coughed, the material wouldn't have gone very far. Then we have the classic at the other end, measles virus. It sends stuff much further. The pathology in the lung makes much smaller droplets that can actually stay suspended in the air, move through HVA systems, move from room to room, get moved around airplanes. It's been thought that the new virus might have been more in this direction, but there's evidence that it actually does things across that spectrum. It makes blobs that are going to fall out of the air, and people have done testing in corners of rooms with patients, swabbed and done PCR for the virus and found it in the corners of rooms. So this is a virus that actually does things that we historically have associated with more this virus or that virus. It's got some properties that go that way. We get to the safety side. I've driven a car a long time. I wear the seatbelt all the time. I seldom need them, but much of the car is designed around safety features that hopefully never actually get invoked. We wear lab coats, gloves, and eye protection. I like eye protection for this virus. I think it's possibly being spread also be microdroplets coming out of eyes, and that's also a target for some infection. Masks, you go searching with Google, you'll find areas of uncertainty and disagreement, but there's a couple of things here. The links are available that mask usage does correlate with reduced rates of infection. So I am a fan of masks. There's argument sometimes who is being protected by the mask. I think they actually provide bilateral protection. Eye protection could be providing bilateral protection. What kind of mask? We're going to hear from Dr. Pearson about that in a few minutes. There's guidance about how to put masks on properly and how to take them off properly and how to avoid contact with the outside of the mask during that process. This is a good video about that. One of the things about us working in laboratories is that labs, for other safety-related reasons, have fairly high air turnover, which is going to reduce virus concentration should someone be in one of our rooms while they're emitting virus. We think about our restrooms in our laboratory buildings, and I think about 10 to the 7th virus genomes per liter of sewage way downstream, mixed with all the other things that go into sewage. And that's motivation to me to go before I go to work and not inhale while I'm in that bathroom if I have to. Small things. So now I get to this. This is my neighbor, Paul. We discuss work-related things, safety and stuff on occasion. He's a PhD chemical engineer, and from his workplace, which is a major chemical company, this idea of layers of protection. And so the outer layer is training. What we're doing today is part of that. Shared values, shared understanding of what the system is, so that you can always visualize, why am I doing this particular practice? Because it makes sense relative to the nature of the thing that we're trying to protect ourselves from. Social distancing away from work. That this is one of the big critical factors in reducing the likelihood of anyone getting infected when you go back into your laboratory building. Don't be the vector. As Ed mentioned, my daughters who are nurses, they need to be careful when they're away from work so they don't take the virus into their patients. Same thing for people in shared workspaces. Don't come to work with symptoms. We have some screening online at Wayne State for that. Make sure you can't get in without doing that, but do be thoughtful when you answer those questions. Some workplaces have the no-touch thermometers. I don't think we're doing that, but at Paul's workplace, they have that. Not perfect, but it's another layer. Surface disinfection, physical distancing within the workplace. This is what we're working on across the campus, is coming up with plans for physical distancing. And during phase 1 of the campus-wide reopening of facilities is reduced population density in buildings to reduce the possibility, if someone comes in who happens to be emitting virus in a transmittable form, that we reduce that likelihood of infecting very many people. Masks and PPE of various forms are typically described almost as though they are the main defense. And my lesson from my neighbor a few days ago, very important one, is that they are the last line of defense. They aren't the first line of defense. And we actually want to set up all of these things so that our mask and PPE is essentially irrelevant unless we failed at some place along the way. It's the last line. It's not the only line. Now, when we think about these, what's the probability of residual risk left? If I do social distancing, there's still some risk left. If I don't come to work with symptoms and I carefully monitor myself, there's still some risk that I'm asymptomatically emitting. All these other things we can think about that way. We can put a probability, what's the residual risk for that? And these, in a layered system like this, the probabilities multiply. And with these numbers, which I made up after thinking about them a few minutes, you could do your own equations. But by implementing these, we end up with a greatly reduced residual risk, I think way lower than going to the grocery store, by implementing these practices in our laboratory spaces. You can play with the numbers and see which variables actually have the biggest influence on the equation in your model of it. But without training and commitment, the restart will crash and burn. Because we control in some manner, not perfectly, but we have control over most of the variables. So going viral, oops. Index case, first person with it, who did they infect? Actually, this one is the index case. They infected 3 people, 4 people, I can't count. And then they start infecting other people. Some don't infect anyone, some infect others. This is a classic virus transmission thing, or how information gets spread in social media. It's going viral. So R0 is the number of individuals infected by each case. So you take the number of people in this, see how many people on average they transmitted it to, count it out on each of those, and you get an R0 for this network. The next 2 slides, I'm going to take slightly slower. Why social distancing matters in the context of reducing R0. So R0 is the basic reproduction number for infectious agents. If it's less than 1, it will disappear in a population. If it's greater than 1, it will continue to propagate through the population. For measles, depending on how it's measured, when it was measured, where it was measured, R0, 12 to 18, that's considered the highest. About 4 are what some estimates are now for the previous virus, the previous SARS, and about 2 for famous viruses, HIV and hepatitis C virus, which are on the infectious side of things. So how do you calculate this? We're not going through all the equations, different ways of doing it, but there are 3 main variables that essentially every model for calculating R0 take into account. The infectious period. This is how long is a person able to transmit the virus. They got the virus, they're asymptomatic, it starts to be emitted, and then it stops after a period of days. That's the infectious period. We don't have a lot of control over this now, but if we had antivirals, we could make this smaller. This can vary, a lot of parameters, age, immune condition of the patient, other things of that sort can go on. Individual lifetime, individual viruses can differ widely. Herpes simplex 2, sexually infected, sexually transmitted disease that comes and goes, reactivates, goes latent, comes out again. It can actually be transmitted over the course of a lifetime of an infected individual. Some viruses, they come and go within 24 hours and then they're eradicated. How often does someone who has the virus and is in a transmittable form come into contact with a susceptible individual? This is something we can control. Social distancing reduces R0 by reducing the possibility of having contact between a susceptible individual and someone who is in a transmittable state. Vaccination, get a lot of people with immunity to the virus, reduces the targets. Large numbers of individuals who have had the virus and have protected immunity. Those things make it so we have reduced the rate of contact with susceptible individuals. We reduce R0. What's the mode of transmission? We have some control over this. We can regulate behavior in our close personal interactions for herpes simplex virus, and we can do things to reduce the dispersal of virus by coughing, masks, coughing that way, those sorts of things. So we have some control over those variables. It's a complex number. By the time you get into the various ways that you can define each of those variables, I think I'm getting at the limits of my time here. If you're really into this, this will be in the slide set that will be available. But the punchline is that R0 changes as variables such as the susceptible fraction of a population change. We can influence this. We can influence by our behaviors, by development of vaccines, things of that sort. Diagnostics, it's a virus that has a genome made of RNA. We can use reverse transcriptase PCR, even in a quantitative mode, and detect the presence where the virus is or was. We can use antibodies against viral proteins and detect viral proteins. That's not widely used for very many virus infections. There are some point of care things that might be useful. Ultimately, we can detect who has antibodies against the virus, who has developed antibodies. After you've been exposed, it takes your body about 2 to 3 weeks for your immune system to develop meaningful levels of antibodies against the virus. Those are fairly easy to measure and inexpensive to measure, and they tell us who had the virus. It doesn't tell us exactly who has the virus. It tells us who might have protective immunity. It tells us who has antibodies in their blood that might be taken out through blood donation and take their plasma or serum and use that to protect someone else. There are also some potential downsides to having protective immunity. There are some viruses, dengue virus, some of you may have heard of in your childhood, that antibody-mediated enhancement of their pathogenicity can happen. The antibodies from your first exposure can then interact with virus particles during a second exposure and lead to kidney problems, amongst other things. So, disinfectants. Remember, the virus doesn't grow on surfaces in the laboratory. It does not replicate outside of living mammalian cells. It's not a bacterium. That's to our advantage here. It has an enveloped virion, so things that can disrupt lipid bilayers can disable infectivity. I have some information behind the screen here, but Dr. Pearson is the expert, and I always defer to him in such things. Treatment and prevention, self-defense, our immune systems actually do a lot to protect us. Self-defense plus support, sometimes in the hospital, keep us alive while our defense systems come on, keep us so we have oxygen, prevent blood clotting. We've got antivirals in development at Wayne State and many other places, vaccines in developing, and prevention, social distancing. We want to not overload the hospitals. In addition to not bring stuff into our reopening laboratories, we need the social distancing to not end up getting the hospitals overloaded again. Detroit had it bad there, obviously, a few weeks ago. Some populations are very high risk, and we need to protect them through our behaviors, people with compromised immunity. So are we safe with respect to the virus? The risk in our laboratory is directly related to our behaviors outside the laboratory. We have imperfect, imprecise predictors of risk, and random happens. We have opinions, there are facts. It's important that we make as many of our judgments on the basis of facts and recognize when opinion is factored into that and how is it factored into that, and how do we modify our opinions as new information becomes available. Within laboratory groups, within departments, safety questions, safety concerns can be turned into useful homework. Group discussions within labs. There's literature, there's Google, we have PubMed access. You can consult other people that might know things. We've got to answer safety questions in a data-driven manner. And then let's remember OEHS and the good people there, Richard and his colleagues, are our friends. This is the last slide. So the way forward, it's new, it's highly transmittable, it's highly pathogenic, it moved internationally in a matter of weeks. Right now, only 1% of the US population is infected, meaning 99% of us are still susceptible. Vaccines take a long time to develop. Hard to imagine actually having one in play that's effective and appropriately tested in less than 2 years, and that would be actually remarkably fast. We need to be able to get quick results for PCR. We need to have serosurveys done to understand how many asymptomatic cases there are, what the prevalence of exposed and hopefully protected individuals are. People are working on that locally and elsewhere. Before we ramp up full around the country and every economic situation, we need to have personal protective equipment, medical equipment, even toilet paper pipelines need to be refilled. I was looking last night at Amazon and numerous brands of toilet paper still not available. Clinical staff, hard-hit areas need breaks. PTSD, just plain old fatigue. Those people need a break before things get ramped up again. And hopefully, antiviral regimen, potentially use of pre-immune serum, might provide some buffer while we develop more robust, long-lasting solutions. So with that, I thank for no one throwing anything. I appreciate the opportunity to speak here and happy to turn this over to Dr. Pearson.
Well, thank you, Dr. Pellett. That was very informative. And I'm going to allow people to unmute their microphones if they want to and clap. And I'm going to clap because this was a great presentation. Okay, so Dr. Pearson, you're on. Okay, I think you need to unmute your microphone, Richard, and I don't think I muted it. Did I mute it? Not seeing you. Okay, there you are.
All right, hopefully you can hear me now.
Yeah.
Now, are you seeing my slides or the?
Yes, yes, you're on with the slides.
Fantastic. All right, well, I'd like to start by thanking Dr. Bhagwat for the invitation to come speak to the students and postdocs of the chemistry department. My talk, like Phil's, was really engineered for their purposes. I'd also like to thank him for the generous introduction of my virology pedigree. And just kind of reiterate that I am here today kind of speaking as a representative of OEHS. And I do appreciate Phil Pellett stepping in here and providing the virology portions of this talk. I learn something every time that Phil speaks. It's very educational. The intent of this talk is to really inform you of the information that OVPR and OEHS has available online, to provide a brief overview of some of these topics, and at the end to invite questions from the students and from postdocs. I'm joined by Rob Moon, who's the director of OEHS, and our goal is really to be able to address questions that are raised by the students and postdocs today related to health and safety topics, but also to learn what kind of materials you need are going to be beneficial as research operations reopen. So, as you are all aware, the governor's executive executive order has permitted research facilities to reopen. The reopening is contingent on the institution implementing a number of precautionary measures aimed at decreasing the potential spread of SARS-CoV-2. So I don't intend to go through all of these in 15 minutes. I really wanted to emphasize here that this reopening is not a return to business as normal. There are a lot of procedures and practices that are going to be put into place. And as we move forward, it's important to recognize that each of us are in a different place with regard to the level of anxiety and stress that we may be experiencing about returning to the workplace. So please be considerate of and respectful of the individuals you work with and the space that you're sharing. Wayne State University is in the process of implementing strategies to ensure that the institution adheres to all of these requirements. These may seem onerous, but as Dr. Pellett's already indicated, your individual commitment is a requirement for successful, safe reopening. I also hope that in showing the scope of the measures that are being implemented, that this will go some way to reassuring those of you who still have concerns about returning to work. So where can you actually find Wayne State University relevant information on the reopening process? So there's a great deal of information on the OVPR coronavirus website that Phil's already indicated the link. Here I've highlighted two of the key documents you'll find on this site. And although this talk, like I said, was geared towards the students and postdocs, to all of the principal investigators that are listening in, if you've not already visited this website, then I encourage you to do so and read these two key documents, because there is a ton of information here about how you should be setting up your labs and what kind of information you need to be sharing with your students and staff. OEHS also has a number of documents on our website. The majority of these are educational documents on key health and safety subjects, such as face coverings and disinfectants. In addition, we have a comprehensive laboratory reopening safety checklist. And I'll show you this here. This is a thorough check on the status of your laboratory prior to reinitiating work. So this document provides information on the training requirements, on the SOP development and review, in addition to the actual self-inspection checklist. So many of your laboratories have been closed for 8 to 10 weeks. And prior to initiating new experiments, it's important that you actually check the workplace for any hazards that may have occurred or developed in the time that you've been gone. So this means that prior to coming onto campus, you need to have done training on the new requirements. You need to go online and complete the Be Warrior Safe training that provides insights into coronavirus specifically and the measures that Wayne State University is putting into place. And that's all individuals wishing to come back to work. You have to understand where and when to be wearing face coverings and be able to have a face covering in order to be able to enter the buildings. You have to have completed a daily health check. So making sure that you are not displaying any symptoms related to coronavirus or COVID-19. And this is a matter of going online and actually completing that online document and receiving something that I believe contains a barcode, which will enable you to have access to the building. Prior to coming to work or in the early stages of returning to work, you need to assess what PPE and disinfectant supplies you have on hand. So what is already available in your lab? This is not only for protecting yourself against coronavirus, but also what is the standard PPE that you have to wear to perform your daily routine tasks in the lab. Supplies are limited and it's harder and harder to try and get some of these key pieces of PPE. So you need to start thinking about what the alternative options are, what alternative suppliers might be, and what can Wayne State University supply to you. There is a stock of disinfectant and some PPE available through OVPR. And then we encourage you to review laboratory protocols and procedures. Some of these may be new SOPs. These must be developed by your lab. As Phil indicated, these can be great homework projects. Spread the wealth in terms of the work that needs to be done here. But everybody should have an understanding of what cleaning and disinfecting protocols you have for your specific lab space. You might want to revisit your work alone policy. We're all encouraging social distancing for safe work. But there are certain experiments and procedures where it's actually encouraged that there is somebody that's overseeing the individual doing the work, making sure that they are safe throughout the entire process and not working alone. So you may need to revisit how that can be done safely in the time of COVID-19 and social distancing. And I know in the chemistry department, you're fortunate that a lot of your office space is actually adjacent to your laboratory space, and you actually have windows between the two. So it could be very possible, in your building at least, to actually have somebody observing from the office space and maintaining social distancing, but also maintaining that requirement to have more than one person present for high-risk activities. And then review your existing lab SOPs. It's been a while since you've been in there. What needs to be revisited and re-examined in the light of this new hazard that's present in your workplace, as well as just refamiliarizing yourself with your general SOPs of how to work safely with the hazards related to your research. And then finally, identifying and resolving issues. So there are a number of things that you need to go through. That's where the checklist comes in. It's a comprehensive list of all the different things related to biosafety, chemical safety, rad safety, waste, etc. But the first thing you need to do is survey your lab for unsafe conditions. What might have developed in the time that you've been gone that you're unaware of? Are there any equipment alarms going off, and what does that mean, and what's the resolution that's needed? Check your emergency equipment. In particular, concentrate on your eye wash stations. These may have been stagnant for the past 10 weeks. We recommend that you flush these weekly for 1 minute. So when you return to the lab, I'd recommend just turning all of these on and letting them run for 3 to 5 minutes to ensure that there's no particulates and material that have collected in the eye wash. Check your chemical storage areas for expired chemicals, your hazardous waste, for anything that might be bulging or over the year-long period that we're allowed to have waste on site. And then review equipment operation safety. Many of you shut down equipment and important pieces of equipment when you left the lab. Many of you may not have actually been through the startup procedure for those pieces of equipment. So make sure that you review the manuals, make sure you start them up safely, and make sure that everything is working correctly, that any hoses or connections haven't frayed or become worn before you start that equipment back up again. So the safety is everyone's responsibility. This includes completing the new online training modules, and the OHS mantra here of safety is everyone's responsibility holds true in these circumstances. All of you must understand the routes of transmission and how the safety measures mitigate the risks, and then implement these practices. So you really do need to understand what you're dealing with, why you're performing certain tasks, and then actually follow through and do them properly. Don't cut corners is the key piece here. So Dr. Pellett's already described the main methods of transmission. We have the primary method, which is direct person-to-person contact leading to others becoming infected. We also talked about, Dr. Pellett talked about contamination of surfaces and objects, either through the droplets forming on those work surfaces or from contaminated hands touching those surfaces. Ultimately, there are a number of points where we have the ability to control the spread and transmission of the virus. So each of these circles represents a point at which we can actually have an intervention. That intervention may be use of face masks, either disposable or cloth face masks, or the use of tissues, or coughing into your elbow, as Dr. Pellett suggested earlier. Another key piece is remembering to wash your hands. This must have been drilled into you a million times in the past 10 weeks, I would imagine, but washing your hands effectively is important to stop the spread of SARS-CoV-2. We'll be talking a little bit about the use of disinfectants and cleaning procedures in the facility for those areas that have become contaminated. And most importantly, remembering that social distancing is key to all of this. You must try to remain at least 6 feet away from your work colleagues and other people in your environment. And it can't be emphasized enough that these strategies are required to be used in combination. There's not one thing that you have to adhere to. You must be using each and every one of these practices to help reduce the spread of SARS-CoV-2 within your building. So, take the necessary precautions to protect yourselves and others. This starts with completing the daily health check form online. If you have any symptoms, please stay home. That is the bottom line. This is not a time to be a hero and try to get work done. And to the supervisors and lab heads that are listening here, please reinforce this with your staff over and over and over again. It requires symptomatic employees to stay home. OVPR has put together a daily return to work checklist. There's a lot of new pieces here that you need to remember to do before you even leave your house, whether it's training, whether it's remember to do that daily health check, remember your cloth face mask, etc. So that's been put together and is available online through OVPR. So staying home whenever possible, and especially if you have symptoms, is the easiest form of social distancing. So only come to work to do the activities that need to be done within your laboratory. If it's computer-based and it can be done at home, stay at home. When coming to work, try to avoid crowded places. Try to maintain a distance of at least 6 feet. And if a building or entryway is crowded when you arrive at work, stay outside and wait until it is clear. And if it's too crowded, maybe just come back at a later time. Departments and lab heads should be working to develop strategies to ensure that social distancing can be observed in the labs as well. So for an example of this is shift work. There could be a strategy that's implemented to try and prevent more than one person or two people being in a lab at a single time. So hopefully this slide is self-explanatory, but just in case, cover your mouth and nose with tissues or elbows when sneezing and coughing. Avoid touching your eyes and nose and mouth with unwashed hands. These are the points of routes of infection. Follow hand washing guidance. Again, 20 seconds, use soap and water whenever possible. And very importantly, and I think a step that most people seem to overlook, is to dry your hands effectively when you complete that process. All of this can reduce the spread of the virus. And if there is no soap and water available, then resort to hand sanitizer, and that should contain at least 60% alcohol. So face coverings is a topic of much discussion. When, where, how, bilateral protection or just individual protection or community protection. Our stance on this is that you're really wearing it to provide others to protect others from you. It must be worn in public places. So if you're sitting in your private office and nobody else uses that space and you're on your own, it's okay to not be wearing a cloth face covering at that time. It's important to reiterate that this is not a substitute for social distancing. Just because you're wearing a cloth face mask doesn't mean to say you can start gathering in groups, in communal areas, etc. Face coverings limit the distance that respiratory droplets can travel, but they don't necessarily protect you from inhaling those respiratory droplets. So also remember that you could be asymptomatic. So even though if you're feeling healthy and you're jumping around and running marathons in your back garden, you could be asymptomatic and you could still be spreading it. So a face cloth is still required. Please also try to wear these face cloths correctly. This is a set of images that I got from the New York Times. First of all, please wear it on your face. The chin and neck is not a suitable or appropriate storage location for your cloth face mask. Remember, this could be becoming contaminated, so you're just spreading this all over your face when you're moving the face mask into other areas of your body. The face mask must be tight-fitting. You can see in the second picture here that it's loose around the edges. There should be a tight-fitting mask around the nose and around the edges. Your chin must be covered, as must your nose. We see a lot of this when you go to the grocery store, seeing people with it just tucked under their nose. This isn't helping at all. And then in addition, it should actually be not just over the bridge of your nose. It really needs to be on the tip of your nose. It needs to be on the bridge of your nose, sorry. So this person is wearing their face mask appropriately. Okay. So we have an OEHS fact sheet. I encourage you to go and check that out. There's a lot of information that's available online about donning and doffing, that is putting on and taking off your PPE. This is true whether you're working in a BSL-2 facility, and now it's true for when you're talking about your cloth face covering. You don't want to be touching the outside of that face cloth any more than you want to, and you don't really want to be touching the inside, so you should be taking it off over your ears. And if it's a cloth face mask, it can be laundered. If it's a disposable, then throw it out into the trash. And another alternative that has been used because of the limitations is to actually just store the masks between uses for 4 to 5 days. And this is not ideal, but in the face of the shortage of PPE, a lot of the regulations related to the use of face masks and respirators has been changed to try and address the shortage. It's important to reiterate that cloth face coverings are not a substitute for PPE. If you are working in a lab and you have a requirement to be wearing a medical grade surgical mask or an N95 respirator or a more a half face or a full face respirator, you can't substitute a cloth face covering for that purpose. And it's also important to remember that wearing a cloth face mask or a disposable face covering is not appropriate in all situations. If you're working with any kind of machinery, you don't want something that could become trapped in that machinery. If you're working with flammable materials, you might want to look into getting a flame retardant rated face covering, or alternatively, in practice, social distancing to ensure that there's nobody else around you so that you can remove that cloth from your face when you're doing that work. And then finally, a disposable face covering is required if you're working in a BSL-2 facility. Just a reminder that any PPE that's used in a BSL-2 facility must remain in a BSL-2 facility, and therefore you would want to have something that you can throw away at the end of that shift. Okay, we're going to talk about the final intervention here, which is cleaning and disinfecting. So cleaning and disinfecting are not the same thing. The words are used interchangeably, but cleaning refers to the removal of dirt from the surfaces. This is important as surface contaminants such as organic matter can limit the effectiveness of active ingredients in disinfectants. While cleaning can reduce the number of microorganisms present on the surface, disinfecting is the active use of a means to kill or denature the microorganisms present. So remember that cleaning a surface may be a requirement for effective disinfection, but they're not one and the same thing. And in many situations, you're going to do both actions. You should also evaluate your workspace for items that cannot easily be disinfected. Your laboratories hopefully do not contain cloth chairs, wooden furniture, etc., that are porous and would soak up any spill, any particulates that fall on them. But your offices might. So these items, such as cloth chairs and the wooden furniture, are actually more challenging to disinfect. And it may be easier to simply remove items that cannot be disinfected from your immediate work environment, so that you surround yourself by things that can be disinfected at the end of each shift. So other considerations, but kindly defer to me on this. We do have, again, a really effective and more detailed document online on the OHS website about how to apply and use disinfectants. But the most important piece here is that the disinfectant that you use must be EPA registered, and it must be on the EPA's list for use against SARS-CoV-2. And that list is provided at this website. And you must follow the directions for the contact time and PPE requirements for that specific disinfectant that you're using. You must address high touch points at least twice a day, probably more frequently depending on equipment use and how many people are using that space, especially if you're doing a shift philosophy. Select a disinfectant that's compatible with the surface of the equipment being treated. Many of you have expensive electronic equipment in your laboratories. Application of surface disinfectants to those pieces of equipment may damage the equipment irreparably. You really want to reach out to your manufacturer and find out what disinfectant type they recommend for use on your equipment, and follow their directions on that. Clean benches prior to use of a disinfectant. We already talked about that for removal of organic load or anything else that may be present that may inhibit the effectiveness of the disinfectant. One thing for you to consider, especially in the chemistry department, is what chemicals have you been using on that bench? You might want to wipe down with water that bench or some other solvent to remove any chemical residue that's present that may interact badly with the chemical that's in the disinfectant. And on that note, do not mix cleaners and disinfectants. Many of these things do not get along. You don't want to be mixing your ammonium compounds with your sodium hypochlorite. So make sure that everybody in your lab is on the same page about what type of disinfectant is being applied to what work surfaces. We don't want one person coming in with a bleach and then 2 minutes later somebody applies some kind of ammonium compound disinfectant. So make sure there's a clear line of communication about what is being used in which area. And then finally, and Phil touched on this a little bit as well on his slide, but household bleach and 70% ethanol are acceptable, but only if an EPA registered disinfectant is not available in your facility. So they can be effective, but they are not tested by the EPA, and therefore they are not permitted as a general means of disinfecting. And if you're planning on doing any BSL-2 work, duly note that 70% ethanol is never accepted as a means of surface disinfectant in your BSL-2 facility. And in general, if you're working with SARS-CoV-2 or COVID-19 patient samples, we're looking for you to have an EPA registered disinfectant in those situations. So once again, if you want more information, check out the OHS website for our surface disinfection fact sheet. And please check out all of our online resources. We plan on continuing to expand this content over the coming weeks, so keep checking back. If you have any questions or concerns or suggestions for additional material, please let us know. The final piece of information on here is the COVID-19 biosafety guidelines. I'm not going to go into those, but I do want to take this opportunity as the biosafety officer to advertise the fact that the IBC exists. This is the Institutional Biosafety Committee. Recently, the NIH Office of Science Policy informed all registered IBCs that we are to consider SARS-CoV-2 a risk group 3 agent. And as Phil already indicated, there's already people that are discussing whether this is actually appropriate, whether it could even be elevated to a risk group 4 agent. Now, typically at Wayne State University, we would not be able to perform any research with this agent due to the requirement of BSL-3 facilities, of which we do not have any. So this changed, though, when the CDC published their interim guidelines for laboratories, and they have decreed that certain activities can be performed at BSL-2, and they use the phrase with enhancement. Some of you may know this as a BSL-2 plus. This is the practice of taking BSL-3 practices and procedures and implementing them in a BSL-2 environment. Now, what enhancements means to your lab is going to be very lab specific, procedure specific, and specimen specific. And therefore, you know, when people keep asking me what enhancements they need, that really needs to be determined by a careful evaluation of the procedures that you're doing and the work that you're planning on performing. So for all of this work, though, Wayne State University Institutional Biosafety Committee approval is required. You must have the approval before you receive the samples. We are working on getting an expedited review process in place for SARS-CoV-2 related research. But I will note to you that one of the biggest hurdles here is that if you need IRB or IACUC approval, one of the things that slows down the approval process in their areas is that you don't have approval from the IBC. So just to reiterate, the IRB and the IACUC will not approve a protocol if it's deemed that an IBC protocol is also required. How fast can we turn these around? That really depends on the quality of the initial application, and it will also depend on the number of protocols we receive. So currently we have had a number of SARS-CoV-2 protocols submitted, and we are turning them around as quickly as possible. But I know that it's never going to be quick enough from the research point of view. But we are trying our best, but we are focusing on making sure that the work is done safely. IBC protocol submission process is performed using the E protocol. We have our own module for IBC applications. This is the same process that you use for any IRB or IACUC submissions. I want to emphasize that a new protocol is required for SARS-CoV-2 research. So if you're an IBC holder, you cannot just submit an amendment to an existing protocol. This needs to be a new standalone protocol for SARS-CoV-2. There is a lot more information available on the eProtocol system through research.wayne.edu on the eProtocol website. This is generic information. Information about the IBC applications can be found on the biosafety website under the OEHS heading. So OEH operations are in place as you reopen. Wherever possible, we will follow the rule of social distancing. So if work can be performed remotely, it will continue to be performed this way. Services such as the pickup and drop off of waste containers will continue, and OEHS staff members will continue to respond to any emergencies or spills on campus. And this is an important point when you guys are going through your self-inspection checklist as you reopen your labs. If you find problems, if you have issues, then both the biosafety, rad safety, and the chemical safety staff are all available for consultation. A quick note on chemical fume hoods and biosafety cabinets. We service each unit annually as required. The closure has put us behind schedule, and we are aware of this. We will be prioritizing efforts as we get back on campus. The primary focus will be the recertification of biosafety cabinets used for processing SARS-CoV-2 research materials. We ask that you work with us as we address all other activities and be patient as we try to work through the process. Finally, for all of our on-campus activities, we kindly ask that you respect our staff and allow them the space to work while socially distancing. So if you guys need waste pickup, please ensure that you move out of the way, get as far away from the folks that are coming in to actually service the laboratories. And that's the same for the staff that come in to certify chemical fume hoods or biosafety cabinets. We won't take it personally if you're trying to avoid us. So on to questions section. This is really what we want to talk about today. I think it's trying to get feedback from you. But before we start that, I wanted to share these 3 additional Wayne State information sources with you. Some of your questions today may be better directed to these groups. The first is Human Resources. The second is the Wayne State University Campus Health Center. And finally, as Phil highlighted at the start of his talk, the Counseling and Psychological Services Group. And this is a stressful time, so I'll end by repeating Dr. Pellett's opening message, which is don't wait to ask for help. That is true psychologically, it's true from the health and safety group. We're here to service you. We want to be able to help you in any way that we can as we move forward through these very uncertain times. So thank you. So I'm going to turn this back to Dr. Bhagwat for him to moderate the question section.
Thank you, Dr. Pearson. That was very informative, and you and Dr. Pellett make a good 1-2 punch kind of team. So that was a good combination. Before I make this open for everybody to kind of chime in with their questions, there are already some questions on the chat. So I'm going to start with the first question for Dr. Pellett. What are your thoughts on SARS-CoV-2 starting to mutate every 2 months, and could it become a problem for vaccine development?
That's a good, hard question that I can't make great predictions on. It's an RNA virus. RNA viruses, most RNA viruses, their RNA-dependent RNA polymerase does not have error correction capacity, and so they have relatively high mutation rates. It becomes a question of selection processes. The coronaviruses actually have a corrective part of the enzymatic activity. And so it has a lower error rate, which slows down the rate of generating things that can be tested in the environment in different hosts and such. So a few things can happen, and it's just some of the possibilities. It would take a lot of changes to actually lead to immune differences sufficient so that the broad collection of antibodies that would be generated in the course of an infection for all of those to be obviated, for all of the epitopes to be modified. So there certainly can be some of that, but my guess is this is a sufficiently complex virus particle, enough proteins on it, big enough, complex enough proteins that there are going to be a number of epitopes on those proteins that mutating one of them away isn't likely to cause immunity to disappear. Another possibility, and this is an interesting one, I think, is that this virus, it's very pathogenic now, and it was able to spread around the world and in cities on this continent very rapidly in its early stages of getting released into humans. And now humans have gotten a bit wise to it. Maybe not hyper wise yet, but starting to change our behaviors. And it turns out that viruses can evolve, and there's some evidence of this in fish viruses that I'm aware of, that they can become less pathogenic in order to enable them to survive in environments where they don't make contact with each other as often. So as we increase social distancing and make it harder for the virus to transmit from one individual to another, there could be an adaptive response where a virus is selected that's actually less pathogenic. So it isn't all bad in the evolution of viruses. I can't predict exactly what will happen though.
Thank you, Phil. The second question is about the lasting immunity. So once you are infected and survive, what is the likelihood that you would survive a second infection?
So here all we have really are historical things to go with. And for the vast majority of virus that I know of anyway, I don't actually know something that doesn't run this way. You get the exposure, you develop protective immunity, and that immunity can persist for a year, sometimes more. If we have seasonal things and you get re-exposed, then that can boost immunity, stimulate memory B cells, and make more antibodies. So I guess I don't have a great answer for that, except to model on what we know about other viruses, that we will get immunity. It will take 2 to 3 weeks to develop. It can continue to increase for a little while after that. And then at some point, having gone up, it will wane and we become susceptible again. But if you get exposed during that waning immunity, but still some protective immunity, it actually, basically, that could be essentially a vaccine for you to boost your immunity without necessarily causing disease. Many different outcomes.
Okay, the next question is for Dr. Pearson. Is there an alternative method for cleaning cloth face masks? For example, using boiling water or something similar, so as to avoid doing laundry every couple of days? Very practical question.
So with the N95s in hospitals, what they've been recommending as a way to kind of preserve is to actually just simply allow time to do its job. And so they've been recommending that you take essentially a brown paper bag, you hook one of the hooks over the top of that brown paper bag handle and let it hang and just let it sit for 4 to 5 days. So if you have enough cloth face masks, you could just simply recycle them through that way. I would still recommend a periodic washing of the entire lot, because you have to remember that you're not just breathing. This isn't just COVID-19. You know, you're out in the environment. You're going to be collecting all kinds of other environmental pollutants and debris on that cloth face mask. So that is a method that could be used. I don't know about, I think on here it mentions boiling water, etc. I don't know that that would be any easier than throwing it into a laundry machine. And I think the addition of the detergent is a plus. So yes, there are alternatives. The use of N95s in hospitals has explored many of these things, but many of these solutions are not going to be available to you as in your home. You know, UVC and etc. is not something that you're going to be employing on a regular basis. So time is one option, but I would still lean more towards doing the laundry on a regular basis.
Okay. The next question is about disinfectant to be used in the BSL-2 facility, because you said ethanol is probably not recommended.
Yeah, so I mean, that's a pretty wide open question. It's hard for me to say what's, because are we in the context of SARS-CoV-2 or are we in the context of some other research? But basically,
I assume good.
Yeah, the bottom line is that 70% ethanol is never a really effective surface disinfectant. And when you consider moving into a BSL-2 facility where most of the work is done in a biosafety cabinet, the airflow patterns in the biosafety cabinet cause that the alcohol to evaporate more rapidly. So whether it's ethanol or isopropanol, they do evaporate too quickly to actually maintain surface contact for the required amount of time to be effective. So for many viruses, if you took a pot of, a beaker of ethanol and soaked it, it would be effective, but not as a surface disinfectant, and that's the difference. And so if you're working in a BSL-2 facility, the consideration should always be, is the disinfectant effective against my agent of choice? That's true for SARS-CoV-2, and it's true for everything else that you may want to work with. A broadly acting disinfectant is best if you're working with blood samples from COVID-19 patients, because it's important to remember that SARS-CoV-2 is not the only thing that's potentially present in that blood. There could be HIV, HBV, HCV, etc. You could be dealing with a whole host of different pathogens that you want to be dealing with. So a broadly acting disinfectant that's good against a wide range of pathogens is what you're looking for. And the other piece is actually a practical implementation. What is the required contact time? Some of these can be as short as 30 seconds, others are 10 minutes. And in general, you'll get better adherence to your cleaning and disinfecting policies if you have a shorter contact time requirement. I know very few people that are patient enough to sit at a biosafety cabinet for 10 minutes, making sure that all the surfaces are wet for that period of time. But if that's what you have available, then that is what is required, and that's what you need to be adhering to.
Okay. There's a question about changing masks, whether you should be changing masks going from the lab to the hallway to the bathroom.
Yeah. So I think this period of COVID-19 pandemic is a really silver lining, is that everyone's going to actually understand what it is to do a risk assessment in your lab. This should be part of your daily life now, I guess, in terms of when you're doing the groceries, whether you're doing laundry, etc. In the laboratory, you need to build this in as part of your risk assessment. What am I doing that may be contaminating my mask? If you're doing something that where you actually may end up with a chemical or particulates on your mask that are related to the hazards in your lab, you need to be putting into place preventative measures to protect yourself from getting contamination there. So I would recommend, instead of using a cloth face mask in that situation, you practice social distancing if possible to remove the need for wearing a face mask. If that's not possible, look for disposable face masks that can be discarded. If they're going to absorb any of the solvents that you're working with, you don't want to have that coming home with you. Other PPE may be effective. If you use a face shield that goes over the top of your cloth face mask, that will protect it if you're working from something that maybe is just a splash issue and hazard. I know that Phil, during his talk, talked about the hierarchy of controls. That's something we always talk about in safety. We have elimination. Can you eliminate the hazard? And then you work your way down. Top of the list is engineering controls. If you can engineer out a hazard, that's the way to go. And you all have chemical fume hoods or biosafety cabinets that are designed for that purpose. So, if necessary, use the chemical fume hood more than you may have typically done for certain procedures if there is a potential for contamination of face masks, etc. So these are all considerations I think you need to start to put into place. During the presentation, I talked about use in a BSL-2 facility has to be disposal. If you're using a mechanical equipment, the last thing you want is something dangling from your face that might get trapped. So I think that there is a role here for you all to be doing a risk assessment of the procedures that you're doing. If you're wearing PPE that could become contaminated, then yeah, it should stay in the lab. So you may want to have a cloth face mask to get to work, and then a disposable face mask or something else in the lab.
Okay, there's a general question here. In case of some other event, such as a chemical spill, would still OEHS come and assist in the cleanup?
Absolutely. So the first thing you'll do, though, is call the Wayne State University Police. So that is the standard response to any emergency. The Wayne State University Police have the emergency responders from OEHS on speed dial. They contact us and we organize our response, depending on which personnel are available. but absolutely OEHS will still be responding to emergency spills.
Okay. The next question is also about face masks. So again, Dr. Pearson, is wearing a mask at all times required? For instance, if someone is working in a lab alone, are they required to wear a mask?
I don't believe so. I mean, this really gets back to whether you believe it's offering you any protection as an individual from aerosols. And I know Phil indicated there was bilateral protection. Our general take is like surgical masks, medical masks in general are really designed to protect your mucous membranes from a direct splash rather than they are from any kind of inhalation of aerosols. If you're worried about particulates, then you really need to be wearing an N95 or a respirator for that level of protection. So if you're alone in a room, there are certainly circumstances where it's perfectly fine for you to take that mask off. And as I indicated, if you're in a laboratory doing work that actually, where the mask actually represents some kind of hazard, then you definitely want to consider alternate ways to be able to perform that without the mask.
Okay. There are about 3 very detailed questions from Shan Hickey for Dr. Pellett. I will just read one of them as there are several additional questions. So the first question is, will the CDC's work on creating an annual flu vaccine, which was decided on in February for the upcoming flu season, be affected by A, lack of workers to create the vaccine, B, bad or missing data because of the COVID-19? I'm not sure that's something you can answer.
I can guess. So my guess is that will continue, and the vaccine for the fall is in production right now. Would be my expectation that workers who do that, facilities that do that, would have been deemed essential, and that would be ongoing. A lot of the information about what strains are included in the vaccine that will go into arms in the fall was collected back in January, February and such, and the final decision usually gets made sometime around in February in terms of what the vaccine would be. And if it hadn't been made by the time things had been shut down, those people would have met by Zoom, something like that. So I'm sure that the decision was made based on customary data for the upcoming fall, and that production is proceeding in a fairly normal manner.
Okay, I'm going to skip the other questions by Dr. Hickey. I suggest, Shan, that you email Dr. Pellett because there are several additional people who have questions. So the next question for Dr. Pellett is, does sewage contain active COVID virions or just degraded genomic content? Is there
That's a good question. And that would require a BSL facility to actually attempt to culture and see what's happened. I haven't read literature related to anyone trying to culture it out of sewage. I think it's probably, at the very least, some of that material is inside of virions. Whether they are still infectious, I don't know, because RNA is fairly unstable chemically in many environments, and I would expect that if it was naked RNA, that it would have been digested.
Okay. A question about OEHS. Does OEHS have a large library of disinfection scopes available so we do not have to invent our own? Much more efficient for the experts to provide them, and labs just choose the relevant scope for the particular need in their lab.
So the answer is no, we don't have a large library of SOPs available for disinfectants. What we do have is a number of different disinfectant documents that direct you to make the right decisions for your lab and your environment. It's very hard for a OEHS to actually really envision every single usage and potential practice in the entire university. So we provide kind of the broad outlines of what it is you need to consider in your workspace. What are you working with? What chemicals do you have present, etc. And whilst we have taken steps to try to provide more than one document on this topic in terms of just both the information on disinfectants, but then also on the application and use of those disinfectants. It's a little hard for us to be able to provide more than that really kind of addresses everybody's needs. But we're certainly here to talk to people, and we'll help people and advise them as best we can if you need information beyond what's included in those documents.
Okay. Phil, this is a question about why measles or COVID-19 virus stays in the air longer than Ebola.
That's a good question. So I think the answer is that just the nature of the pathology in the lung with those viruses results in smaller droplets being formed in the course of the coughing or whatever would happen singing with the new virus that get the virus emitted. And then it's a function of them being sufficiently small to float in air. And with Ebola, they're bloody clumps of bloody lung secretions that simply fall out of the air more easily. It's a particle size thing.
Okay. There's a question again about masks. That seems to be the most popular topic. What is the final treatment given to the used mask discarded at Wayne State? I guess, how do we get rid of them or make them not infectious?
If they are disposable masks that are being used at Wayne State University, it's a great question because I hadn't really thought about it. But I would probably at this point say that many of you have the biohazard bins available. This would be a perfectly appropriate use for you to get rid of that way. They would be autoclaved out by our contracted vendor, Daniels. But in reality, you know, the cloth masks that you're bringing home are probably similarly contaminated. So I don't think there is a huge risk here. And if they ended up in the regular trash, I don't know anybody would be terribly disgruntled. The one thing I would say that if you're in the lab, think about what other contaminants may be on that mask, and that may actually influence your disposal route as well. So anything that's acutely contaminated should be, you know, whether it's chemical, radiological, or biological, should be following the appropriate waste stream for that particular hazard.
For example, in the chemistry, there are many labs that have the autoclavable bags and everything available. That's probably easy to see discarding there. But in a physics department, there may not be such things, and maybe the OHS should think about what they should do.
Yeah, we can certainly do that, whether that's just maybe providing a couple of bins per floor or something for disposal, right, or whether, you know, we determine that they're okay to go in the regular trash. I think that's actually a really important question, and I'll certainly try to give that some more thought and consult with our with our hazardous waste team and see what kind of solutions that they can provide. But I think that's a great question.
One last question in the chat is that you said that no mask is needed. I'll wait for you to make your notes. Yep. Yeah, you ready? Yeah.
Yep. Just writing down the last question so I don't forget.
Yeah, sure.
Go ahead.
So you said that no mask is needed if working alone, but that is not consistent with the chemistry department policy because we are never recommended to work alone. So just
I think that, you know, it is consistent with the university's policy, as far as I'm aware, that if you're alone in a laboratory or you're alone in a office, that the mask is not a requirement. Any department or group is welcome to make you can be more strict than what we decree. We tell you what is required and what the regulations state. People can always be more conservative. What you can't be is less conservative. So that is open to interpretation by individual groups if they want to take extra steps to ensure the safety of their people or feel that it's necessary to take those extra steps. So I would just say that yes, if it's coming from OEHS, be more conservative is fine. Being less conservative is not appropriate. And like I said, I believe that this is consistent with the university's institutional policy as it stands.
Okay. I think I made it such that people can unmute their microphones. So if you have questions, welcome to unmute your microphone and ask the question directly, or keep typing. But in any case, if there are no more questions, then I would from the bottom of my heart, would like to thank both of the speakers. And so maybe everybody can unmute their microphones and give a round of applause.
Thank you very much.
Thank you.
All right. Thank you.